Question 4, and we've got some contents of a cell, a generalised animal cell, and hopefully some things that you recognise in there. And what can we see? So we can see this looks like a half of a nucleus with nuclear pores. So the blob inside the nucleus, that's the nucleolus. E is chromatin or the DNA. We have got F and C appear to point to the same thing, but C the little dots and F is the membranes. So F is rough endoplasmic reticulum, C are ribosomes, B is the Golgi body, A mitochondria. They're both the same thing, but one is kind of facing towards you and cut sideways. Some of these organelles are involved in the functional protein as shown in the flowchart below. State how B to F work together in this particular sequence. So B, C, D, E, F. So not necessarily the same order that it's mentioned in there. B to F. We're probably best going along with this flowchart. So E, there's no marks for saying what the thing is. So no marks what we've put so far. It's how they work together. So E is the DNA, so that carries the genetic code, and that's what's going to get transcribed onto messenger RNA. So transcribed onto messenger RNA. So later on you'll do about messenger RNA. But if he carries the genetic code, is enough for today. Then what are C? So C, the little dots there, ribosomes. So C, ribosomes, and that is, well, later on you'll say translation of messenger RNA to form the polypeptide chain. Or probably for today, if you're saying put some amino acids together into the polypeptide chain, so amino acids joined together, that sort of thing, into the polypeptide chain. What does D do? D is that nucleolus, so that's involved in the production of ribosomes. So D produces ribosomes. F, so what are the membrane part of the rough endoplasmic reticulum? Well, that's involved in kind of packaging and transport of the proteins. So packages, transports the proteins or the polypeptide chains, and that's going to then form vesicles and off to the Golgi body, to the Golgi. And then lastly B, that's the Golgi, modifying the proteins. So making them functional, might be adding bits, adding to the structures. So modifies proteins, and you could also say packages. So if you know your cell structure in your organelles, 5 marks there should be okay to go. It's just AO1, remembering stuff. Suggest the role of organelle A. So it's supposed to be AO2 this, but it's just really telling us what the function of it, and then relating it to this process. So there's just one mark, so makes ATP in aerobic respiration. So it makes ATP. What's this ATP used for? To provide energy for protein synthesis. For example, peptide bond formation. If you're building things, you need to put energy into it. Calculate the actual size of organelle A between G and H. So if I put my ruler on, we get 24 millimeters. So the image size is 24 millimeters. And we've got the magnification there, 12,000. So it's not really 24 millimeters, it's 12,000 times smaller. And so it's going to be divided by 12,000. Especially when I'm using the most appropriate unit. Well, millimeters isn't going to be any good, but we're going to have to choose the next one down, which is going to be micrometers. So I might as well convert that now, because it's easier to convert it now. So 24,000 micrometers. And now the actual size is the image size divided by the magnification. So it's 24,000 divided by 12,000, and you can do that in your head. That's going to be 2 micrometers. Organelle A is similar in size, so mitochondrion is similar in size to prokaryotic cells and has structures common with them, and this led to the endosymbiosis. Using your knowledge of cell structures and processes described, justify this conclusion. 2 marks, and you only get two little lines here. So this is AO3, which is using your knowledge to suggest kind of new outcomes to experiments or theories and that sort of thing. Basically, you're just comparing the mitochondria with your knowledge of prokaryotic cells, looking for similarities. So A, which is the mitochondrion, has similar, so for example, small 70S ribosomes. We could also suggest it's got 2 membranes. So this is if there's mitochondrions like this, so 2 membranes, but you can possibly imagine if it was a prokaryotic cell and engulfed by another cell, then you've got those 2 membranes again. So 2 membranes, same as if had been enclosed by another cell. So that sort of ideas. So pretty difficult really to think of lots of things, but other little similarities. Other similarities might be small loops of DNA. So 3 things there. You need 2 of them, which give the kind of similarities that between that mitochondria and prokaryotic cells that would tie in with this theory. So a difficult 2 marks, but you should have got the rest of it pretty easy at the other 8.